Hello, everyone. Welcome back. Thank you for joining us. I'm Steve.
And I'm Scott.
And together we're Backyard Musings, broadcasting live from Apple Valley, California.
And we are still talking science and technology.
Scientists have discovered new markers that enable the identification of species from fossilized bone fragments, offering new insights into the extinction of mysterious megafauna. What happened to all the megafauna? Across the world, from towering mammoths to flightless moas. Are they talking about the moas on Rapa Nui, on Easter Island?
I don't even know what a moa is.
Many of these giant animals vanished between 50,000 and 10,000 years ago. Well, the mammoth did for sure. Direwolves did for sure.
I know what a mammoth is.
Unlocking the mystery of their disappearance could reveal vital clues about ancient ecosystems and help predict the fate of modern species facing extinction today. Would you like to know more?
However, studying these prehistoric giants is not easy. Fossils are often too damaged to clearly identify, and in warm or humid climates, DNA rarely survives the passage of time. Now, a team of scientists has made an exciting breakthrough. They have isolated unique collagen peptide markers that can reliably identify 3 of Australia's ancient megafauna. Now, this part right here gets me. A wombat the size of a hippo. I don't know. I'd have to see it to believe it.
A giant. Go to the tar pits, man.
I guess. A giant kangaroo and a fearsome marsupial with enormous claws.
This sounds very interesting. Stay with us here. Very interesting.
If they killed one of these, they could eat for probably a decade, right?
Yeah. They probably herded them, scared them and run them off a cliff, and then they went down to the bottom and feasted, like you said, like the native Pacific Islanders for weeks.
Why would these things be scared of a man? I mean, they're giant.
Animals.
Hippos aren't scared of
they're
men.
Herbivores. If they're carnivores, they wouldn't. They'd be trying to eat the man.
Okay.
Something trying to hunt them and eat them. It's a critter with natural instincts. Quote, the geographic range and extinction date of megafauna in Australia and potential interaction with early modern humans is a hotly debated topic, said Professor Katarina Duca of the University of Vienna, senior author of the article in Frontiers in Mammal Science.
Quote, the low number of fossils that have been found at paleontological sites across the country means that it is difficult to test hypotheses about why these animals became extinct. This is explained by first author Dr. Is that Carly or Carly?
Carly.
Carly Peters of the University of Algarve.
Algarve. Where is
okay.
That, man?
That's where all the Algarves hang out, I guess. Archaeology by mass spectrometry, MS, could increase the number of identified megafauna fossils, but only if collagen peptide markers for these species are available. Unquote.
Analyzing the peptides, which are short chains of amino acids, like Dino, the little dude on Jurassic Park, the little round dude, the DNA? No. Okay. Found in samples of collagen allows scientists to distinguish between different genre of animals and sometimes between different species. Because collagen preserves better than DNA, this method can be applied successfully in tropical and subtropical environments where DNA is unlikely to survive. But most reference markers are for Eurasian species that never lived in other parts of the world. This research develops new reference markers for an Australian context, allowing scientists to glean more information from Australia's fragmented fossil record.
Do you glean often?
Sometimes.
Do you?
Sometimes. It's a technique that I sometimes use.
I don't know if I've ever gleaned.
You've never gleaned?
I don't know.
Have you ever gleaned?
Quote, proteins generally preserve better over longer time scales and in harsh environments than DNA does, says Peters. Going on with the quote, this means that in the context of megafauna extinctions, proteins may still be preserved where DNA is not. How is that possible?
We're about halfway through, so stay with us, people.
I thought DNA was something that just lived on forever, right? I mean, that's the big...
I guess it could degrade. I mean, it could, rot, right? Couldn't it?
Well,
there's no DNA in
hair
fossil.
Doesn't rot, right? Hair
then we'd have a world full of hair, man. Yeah, it rots and destroys. Yeah. Yeah.
Okay, I thought that's one of the things that
Teeth?
kept growing on a dead body.
Well,
Your
I'm
hair
sure
keeps growing.
I'm sure down in the coffin, yeah, the hair might grow for a little bit.
Oh, okay.
Especially if it's hermetically sealed, like Princess Diana's coffin was, lead and hermetically sealed. All right.
Okay.
The scientists chose to study Zygomaturus trilobus, Palorchestes azale, and Protemnodon mamcura. Woo! I got those, man.
It sounds like something on the menu at the bento
Maybe we'll call
restaurant.
them ZT, PA, and PM. We'll call them that.
Okay.
3 species which could be particularly valuable for understanding megafauna extinctions. Z. trilobus and P. azale are from families of animals that went completely extinct during the late Quaternary, while P. mamcura survived long enough that it could potentially have overlapped with humans arriving in Tasmania. Dr. Richard Gillespie, a co-author, previously dated the bones to beyond 43,000 years ago.
Okay, here we go. Zygomaturus trilobus was one of the largest marsupials that ever existed.
Oh,
okay, so this is the thing that's bigger, the wombat.
Yeah, it's a hippo size.
Yeah, it would have looked like a wombat the size of a hippo, says Duca. Protemnodon macura was a giant, slow-moving kangaroo, potentially walking on all fours at times. Oh, jeez. Palorchestes azale was a unusual-looking marsupial that possessed a skull. A skull. The head was highly retracted nasals and a long, protrusible tongue. I think I went to school with these. Strong four limbs and enormous claws. If the early modern humans who entered Sahul, the paleo continent that connected present-day Australia, New Guinea, and Tasmania 55,000 years ago, came across them, they would have certainly got a big surprise.
What about Zealandia?
Really?
They didn't mention Zealandia here.
We talked
That was
about
a huge
Zealandia.
continent.
We talked about that, yeah.
Huge continent.
Yeah. Refer to our videos.
The scientists ruled out any contaminants and compared the peptide markers they found to reference markers. The collagen in all 3 samples was well preserved enough for the team to identify suitable peptide markers for all 3 species.
We should have some kind of what do they call that when the words go across the bottom of the
Chiron.
screen? Chiron.
Yeah,
we should have that for some of these words that I butcher, that people could say, "Oh yeah, I know what that is," because I know they don't know what it is. By the way, I'm saying
that isn't. There's probably got to be a way to do it, with the editing and stuff.
Yeah. Using these markers, the team were able to differentiate Protemnodon from 5 living genera. That's not genre, is it?
Whatever. Genera.
And 1 extinct genus of kangaroos. They were also able to distinguish Zygomaturus and, oh my gosh, Palorchestes from other living and extinct large marsupials, but they couldn't differentiate the 2 species from each other. This is not unusual with zoo MS, since changes in collagen accumulate extremely slowly over millions of years of evolution. Unless further research allows for more specificity, these markers are best used to identify bones at the genus level rather than the species.
However, the ability to tell apart genera from more temperate regions of Sahul, that's the name of that continent, that supercontinent, does present an opportunity to try to identify bones found in more tropical areas where closely related species, which are likely to have similar or even the same peptide markers, would have lived. DNA rarely preserves over time in these regions.
Quote, by using the newly developed collagen peptide markers, we can begin identifying a larger number of megafauna remains in the Australian paleontological assemblages, says Peters. Quote, however, there are a lot more species for which collagen peptide markers still need to be characterized. 2 examples would be, of course, the Diprotodon, the largest marsupial genus to ever exist,
Ooh.
and the Thylactoleo, the
Sure.
largest marsupial predator,
Oh, the ones that ate the marsupials. Huh.
a marsupial that ate its own.
So if you're down under, you probably find this really interesting.
If you even know these words, we need a what'd you call it? A gyron?
Chiron.
We need one of those.
We're going to work on that. You can probably see in the reflection Cameron swinging on the swing over Scott's left shoulder.
He's a large marsupial.
All right. Thanks for joining us, folks. We'll talk to you later.
Take care, everyone. Thanks. Hey, you did your hair.